Okay, we're live now. It's going to be a bit of a lag. I'm going to wait till it shows up over here on my other screen. There we are. Make sure I'm muted on that side. Okay. Yay! Bring up my notes. We'll get started here in a second, everybody. Welcome. Let's see. Okay, welcome everybody. Good afternoon. Welcome to the Lunchtime Lecture Series from BRIT and the Fort Worth Botanic Garden. I'm Dr. Brooke Best. I'm Director of Research Programs here at BRIT, and I and my research colleagues host this lecture series, which takes place usually on the first Tuesday of every month. Our next talk is actually going to be on Friday, April 9th. It'll be by Dr. Peter Fritsch. The title is Evolutionary Origins and Species Diversity in the Wild Blueberries. But today we're streaming our lecture live to YouTube, and you live viewers, you can type your questions here anytime, and we will answer them at the end of the lecture during a Q&A session. And at that time, your questions will be displayed on screen. If you'd like to test out the chat now, feel free to share where in the world you're watching from. I do want to mention that this lecture and the accompanying live chat are being recorded and will be available in the future on BRIT's YouTube channel, pending permission from the speakers, of course. If needed, links to other resources mentioned during the talk will be posted to the BRIT webpage for this event. And if something happens and we get cut off, just refresh the YouTube page or check the event page at brit.org, as we'll post new information there about possibly reconnecting or switching to a new link. So I'll now turn it over to Tiana Raymond, BRIT's Herbarium Collections Manager, who's going to introduce today's speakers.
It is with utmost pleasure that I introduce our two speakers to you today. I've known these women through the herbarium and botany world, but over the last couple of years, I've had the privilege of working more closely with them, being welcomed into their homes and collection. There's not quite anything like bonding over heavy lifting and herbarium work as we collaborated in the repatriation of specimens to the Tucker Herbarium at LSU from the Ardell Thomas collection, which is now at BRIT. But our first speaker today is Jenny Kluse, the Herbarium Collections Manager. She has a background in plant ecology and conservation with a master's from UC Berkeley. While her family makes an annual migration to the Rocky Mountain Biological Lab every summer, traversing a diagonal line through Texas to sample field plots of plants and lichens, her primary habitat is in the climate-controlled recesses of LSU's herbarium, from which she imagines the former longleaf pine savannas and Gulf Coast marshlands, while filing specimens. To close out this presentation, we have Dr. Laura Lagomarsino, Director of the Herbarium and Assistant Professor in the Biology Department at LSU. She has a PhD in organismic and evolution biology from Harvard University and a BS in genetics and plant biology from UC Berkeley. Her research has taken her on field expeditions throughout mountainous Latin America in search of bat and hummingbird pollinated plants. She is an advocate for the importance of collections and research and diversity in STEM. Without further ado, welcome to the Shirley C. Tucker Herbarium, a past and future hub for Louisiana Botany.
Thank you, Tiana, for that introduction. I am Jenny Kluse, and I'll be giving you an overview of the Shirley C. Tucker Herbarium and our current project integrating divested regional collections, which helps to reinforce us as a hub of botany for the state. And then Laura will be finishing up the talk, describing some of our research projects that are going on. And so we house the oldest botanical collections in the state. Shown here is some specimens that were collected by Thomas Drummond in 1832 when he made an expedition into the Americas. He was a Scottish botanist from a family of botanists, and he started as a bryologist, but he was an incredible collector coming into the States and dealing with all the difficulties of those times. And he made his way from doing this collection in Louisiana over to Texas, collecting both plants and birds for several years. And because of his incredible efforts, some of the species are named after him. You might recognize his name, Drummondii. So this is Nolophia drummondii, which is also called propeller flower or pine woods lily. And it's actually, like common names can do, it's not a lily, but it's in the iris family. And here's a pre-pandemic picture of our herbarium crew, just so you get an idea of how we're structured. Laura Lagomarsino's lab is the core of our research program here, as well as Sarah Matthews' lab, who has been recently introduced to working in the biological sciences. We've been able to add on a new botany professor, which is great. And then we have an herbarium graduate curator assistant. So that's Lamone in this picture here. And then we have a part-time staff. She's not pictured here. Christina Mazzucato works with Shirley Tucker and her lichen collections. And then we've been supported by the National Science Foundation to do some of our integration and digitization programs. So we've been able to hire on some staff members to work on that, like Danielle Santa Maria Aguilar and Jenna Mount, and also Laura Frost part-time at that. And then we also have, you can see a lot of young faces here. There's a big group of undergrads that work with us, and they're supported through either work study, or some of them have scholarships too that helps them to be able to do some work in the herbarium and get some experience here. And lastly, we have a tiny group of volunteers that we draw from the Louisiana Master Naturalist program. So to understand how our herbarium is broken up, we have 5 major taxonomic groups, starting with the vascular plants is our largest portion of our collection. And vascular plants, just as a reminder, have conductive tissue of xylem and phloem, helps it give them the tall stature too. And you can see this includes gymnosperms, which there's longleaf pines in the background here, and angiosperms, including this orchid and rhynchosphere growing next to it, and also includes pteridophytes and lycophytes like ferns and their allies. And so our second largest collection are lichens, and this is the collection of Shirley Tucker. So her research was focused on, here at LSU, was focused on plant developmental biology, but since an undergrad, she was collecting lichens. So it took her all over the world collecting lichens and exchanging material with Australian collectors, European collectors, and she still works on her lichens today. And then here is a non-symbiotic group of fungi. We have a separate collection for our macrofungi. So these are non-lichenized fungi. They don't have the algal partners and cyanobacterial partners that some of the other collections do. And those are, things like big mushrooms. We don't have very many microfungi in our collection, but here's an example of a common stinkhorn mushroom that is hard to miss. So it's bright orange, and it comes up in wood chips underneath the live oaks around campus here. And then another plant collection that we have separately are the bryophytes. They're non-vascular plants, so they don't have the xylem and phloem tissue, which makes them, small diminutive plants. And they're broken up into 3 groups: the hornworts, which are these greasy green looking guys, and liverworts, which we see commonly along the clay banks of some of the riparian areas here in Louisiana. And then mosses, which this is a sphagnum moss, which is ubiquitous throughout the world. And then we do have a small alga collection. It's mostly marine seaweeds, so we house those too. So quite a diverse set of taxonomic groups. And so one of the purposes, or the purpose really, of an herbaria is to catalog life on Earth. And not all life on Earth is distributed equally. We have these different biodiversity hotspots. The Kew Gardens puts out an annual report each year of the state of the world's plants and fungi. So in that report, they've estimated that there's about 391,000 plants that have been described and 120,000 fungi. So that's quite a few, and it's an order of magnitude higher than the number of vertebrate species that are described. Also, they've estimated that about 2,000 to 4,000 plant and fungal species are described each year. So that's part of the work that we do in an herbaria, is to help describe new species. As for comparison, the Shirley C. Tucker Herbarium has about 16,000 plant species and over 7,000 fungal species. And so while that's only representing it's less than 5% of life on Earth of those taxonomic groups, you can see that our distribution is mostly in North America, and but we do represent all the different continents except for Antarctica. We don't have any species from there, but you can see that we have a really strong focus on regional areas. So our vascular plants and lichen collections do a good job of supporting taxonomic groups that are from the Gulf South, and especially Louisiana. And part of this integration project we've worked on with BRIT was to help kind of grow our herbarium to represent all Louisiana taxonomic groups for plants. So there's about 3,200 plant species in the state. And with the wonderful collaboration we were able to do with BRIT, which was a tremendous amount of work. Peter Fritsch supported it, and Tiana Riemann and Ashley Bordelon were incredible collaborators on this project. But we were able to grow our collections to represent the western part of the state as well as the northern part of the state, where we didn't have as many collections. And then we also work with the Louisiana Department of Wildlife and Fisheries, and we're the repository for their collections that continue to come to us. And we're like many other museum or natural history collections. We send loans out all over the world. For example, I recently sent a loan to Belarus, which was to a Russian scientist, and we were not able to send loans through to Russia, but she was able to have the loan sent to a nearby country so she can study some lichens there. And so that way we support research to collectors all over the world. And we have an exchange and gift program that helps us to grow and diversify our collections. In good years, we conduct collection expeditions, and we have visiting scientists. As you can see here, there's an undergrad working to prepare a specimen, and also on the other image, we're demonstrating the botanist stoop, which some of you may be familiar with. Oh, and then the other thing we do is Plant ID Fridays. We have people come bring specimens for identification, or we work on material that hasn't been described yet in our collections. And recently we've been able to grow our outreach program over the last few years. We collaborate. There's a museum on campus that houses the birds and the mammals and the fishes. And so we've worked with them to collaborate on programs such as Special Saturdays or Night at the Museum. We do a citizen science outreach program each year that's part of a larger effort in collections called We Dig Bio. And we give tours of our collections. And we've been very fortunate recently to have some guest speakers, Pam Soltis, who's from Florida, and the world-renowned Lucia Lohman coming to our collections and giving talks. So that's been wonderful. And a new thing that we have is our display case, which is outside the entrance of our collection. So we don't have the museum display right here on campus, but we are able to have a small display case and have a rotating exhibit in there. And our facilities are on the LSU campus. We're housed with the Department of Biological Sciences. You can see here these windows on the second floor that wrap around the building. That's where we're located. So it's a large space where we have our compactorized cabinet systems. And we have a small library and workspaces for visiting researchers. So since 2015, our collections doubled in size. So that was a big change for us. And you can see the different collections that we've integrated, including what Tiana called our Dale Thomas collection that is at the University of Louisiana, Monroe. We were able to repatriate about almost 60,000 plant specimens from that. A year before that, the Tulane University divested its collection, and we were able to work with them and bring the collections here to LSU, and that was 120,000 plant species. And then there's been some smaller collections in Louisiana, like the LSU Shreveport. We have a good size of their collection. The McNeese State Herbarium, all of that came to us. And the last bit of bryophytes from the Lafayette collection came to us, and two other collections from Florida, the Eglin Air Force Base collection, which is mostly plants from the longleaf pine savannas, and then a portion of the Tall Timbers Research Station herbaria, which was a selection of bryophytes, which were part of a fire ecology study. So unfortunately, it's so sad when these collections close their door, and it's not a good thing. It blocks direct access to collections for researchers. So while that's disconcerting, on the flip side, we've been fortunate to be a trusted place to send collections and to acquire these. So we've been fortunate to get these beautiful collections. And I think part of the reason for that is we have a strong online presence where we host our data freely, so anyone can look kind of to see what's inside of our collections. And then also we've had a wonderful administration that's for a long time supported collections-based research. And along with this Walmart size growth that we've had here in our collections, we've had to integrate the digital collections too. So we have the physical specimen, but we've also have digital records of nearly every accession in our collection. So we've grown to nearly 400,000 specimens, and we host the server for the Louisiana images. And just so you know what a digital specimen, digital record is, it's really when we put a barcode or a catalog number on a specimen, and then there's associated data with that. So it might be just a photo of the specimen itself, or it could be data associated with that collection that comes off the collection label. So it would be the scientific name, who collected it, where they collected it, when, and so data can keep going from there. But that's the digitization work that I talk about here. And then when we made this, big jump in size, we ended up switching our database into a Symbiota database, just that made it much more efficient for us to work with multiple people databasing, which was not an easy decision, but we're glad we're now in it. And then also we have some wonderful people working on restructuring our digital data flow, including Jenna Mount, who is a graduate student here. And Jason Best has been a part of that too. So thank you, Jason at BRIT, for helping to restructure some of our data from the NLU collection. Okay, so now I'm going to talk about the three herbaria that are the largest in our collection. As I mentioned before, Tulane Herbarium is 120,000 plants. The 10,000 fungi were earlier moved to the New York Botanic Garden, if people are wondering about those, from Arthur Weldon's collection. The founding collection for Tulane is from the 1840s, and it's from the New Orleans Academy of Sciences, which was a group of doctors that had their training in botany, like a lot of doctors did those days, because the medical sciences and botanical sciences were more closely related, and still are related today, but there was more of a tight connection. And so those collections are the early collections that we have, some of those early Louisiana plants, and they helped to create a checklist of early Louisiana flora that was part of the Torrey and Gray's Flora of North America. And Joseph Ewan, if people don't know him, he was a director of the Tulane Herbarium for many years. He brought his personal herbarium collection of 32,000 specimens. He was also a historian of botany and botanical explorers. So he has some really interesting books. And because of that, he collected really interesting material and was able to get some neat material in exchange and buy gifts, such as that Drummond specimen we saw earlier. And in 1977, for many decades, Steve Darwin and Anne Bradburn were the curators of the collection, and they did a beautiful job of keeping these specimens, which none of these specimens use glue, which is unusual. They're all affixed to the sheet with linen tape. So they're absolutely gorgeous specimens to work with. And then, just so you know, the Tulane Herbarium, we've kept its identification distinct. So it's still the Tulane Herbarium that has the NO herbarium code, whereas the rest of the collections are LSU or LSUM for our fungal collections. And the Louisiana Monroe collection was the largest collection in the state of nearly 500,000 specimens. They're attributed to Ardale Thomas, who had the highest collector's number of over 173,000 specimens that he collected in his lifetime. They, as I've mentioned before, were relocated to BRIT, and we've been very fortunate that they've been working on these programs to repatriate some of the specimens to state institutions such as LSU. And the Louisiana Herbarium, its history starts with America's Featherman in 1869. In 1944, those collections from America's Featherman, who was an early, this is when the university was just starting up, they were brought back from Claire Brown, who was a curator, and they were at the state archives, and he was able to bring them back, which are a lot of our type specimens now in our collection. We also have, like I said, the mycological collection of Bernard Loeys, who mostly worked on wood rot fungi in the tropical Americas. And for another 40 years, our second director was Lowell Urbach, and he worked on Asteraceae. And so we have a lot of Asteraceae representation in the collection, as well as Cyperaceae, and we have a good portion of Rubiaceae too, which is the coffee family, which Steve Darwin worked on at Tulane. So in 2002, our collections were consolidated in the Life Sciences Annex. Before that, they were more people's personal collections outside in the hallways. So we were able to get this beautiful space in 2002 to move our collections in. And in 2015 was another big year for us. We received an endowment from Shirley and Ken Tucker, which is the namesake of the herbarium. And that helped to support a new researcher studying botany here too, which is Sarah Matthews. And in 2017, Laura Lagomarsino came to the collections and as a new professor. And the first day on the job, she was working on trying to get support for integrating all these newly divested collections into this big space. I cannot miss talking about Margaret Stones. She was a botanical illustrator for Q and did the Curtis's magazine for many years. She came to Louisiana to work on a project to illustrate the native flora of the state. So her work can be found in several books now. Their large folio print book just came out from LSU Press, and her originals are in the Hill Memorial Library. So if you get a chance to look at those, they're absolutely stunning and intricate and gorgeous. And as I mentioned, our 5 different taxonomic groups, I just want to show you where to find our, information from our herbarium. They're in 5 different data portals. So the ERNEC is where our vascular plants, then we have our lichens separate from our bryophytes, and our fungal collection is, and then our algal collection also separate. And we do move our data to other data conglomerates such as GBIF, which many researchers use. And just a quick, how to do a search in our collection. If you go to one of these Symbiota portals, you can select all the different herbaria, or you can just select one herbaria and you can get a nice table view, and then you can link out to these little camera icons and get a high resolution picture of the specimen. So you can actually see inside our collections now. It used to be considered dark data. Now we're bringing it to the light. And for our records that have lat-longs or georeferenced points, then you can do a map search. And so you can say, okay, I'm going to this area, what might be found here? And you can link out to collections that have been, this one's collected by Chris Reed, and it's a black mangrove on the Gulf of Louisiana. And you can see all the others that have been collected along the Gulf too. Okay, so just to show you some of the important and fun collections in our herbarium, the most important species are probably the type specimens, because they're when species are first described to science. So as I said, we have a lot of America's feathermans type species. This one is actually at the Harvard Herbarium, and it's unusual because it's a painting of a specimen rather than the physical specimen itself. So I thought that was kind of fun for those of you who are familiar with collections. And it's now Ludwigia peploides, subspecies peploides. But the original name had Boydiana, which was an attribute to the one of the starters of the founders of the University of Louisiana system in the 1800s. Okay, and probably most famous collector that we have, Joseph Banks and Daniel Solander. This is from Captain Cook's first expedition when they were charting the distance from between the Earth and the Sun. So this is collected near Rio de Janeiro, and it's what is it? It's an Ipomea, so it's a morning glory family, and that was attained from the British Museum, probably as exchange to Joseph Ewan at Tulane. And Inez Mexia has made it to being a Google Doodle. She collected prolifically, and she's an inspiration for those who find their passion later on in life. She started collecting and doing botany when she was in her 50s, and she was part of the Sierra Club in California, moving from Mexico, and she just got hooked, and she ended up doing many expeditions into Latin America, and she collected in these remote places in the 1920s and 1930s, most of the time on her own, and she would hire guides along the way to help her move these gigantic, reams of specimens. And but so we have quite a few in our collection, and her duplicates were distributed by the California Academy of Sciences. But this is a beautiful Valosia growing in Brazil. It's like a shrubby plant with a beautiful flower. And I've talked a lot about plants, our fungal collections. We have this really fun zombie fungus. It's a cordyceps. This one's from Brazil, and you can see the ant here growing out of its thorax, has a fruiting body. What happens with this fungus is it co-opts the nervous system of the insect, and it makes it climb to the light. And when it gets to the light, the reproductive body shoots out and spores go spraying around. So that is a fun one. And recently there was an undergraduate working on University of Louisiana Lafayette, and he discovered a queen carpenter ant doing a strange movement on the ground, and it is another it's another zombie fungus basically that he described, and it only the host is just, they think, the queen ant, and once again it zombifies her and she goes above ground and spreads out all the spores. So that's one that is, deserves more ecological studies too. And ultimately, we hope that our work here can contribute to conservation of species. In this case, it did. This is a collection from a former grad student at LSU. She's now a professor in Mexico, and she worked to describe some of the maple species. And this is a cloud forest population that she was able to elevate the name to species status and then show that it really needed to be protected as a critically endangered species on this rainforest, or it's like a cloud forest reserve in Mexico, and where it was threatened by nearby logging. So from here, I'm going to let Laura continue with some of other research that we have going on here at LSU.
Great. Thanks, Jenny, for that first half. I always love hearing about our herbarium. I'm going to go ahead and share my slide here. Hopefully you guys can see that. And Jenny's discussion of Yalma's research is perfect translation to talking about the research we currently have going on at LSU. Jenny talks about the curation we do, but we do a lot of research here as well. I'm going to start this half of the talk talking about the extended specimen network, this concept of herbaria being, and other types of natural history collections being, not just the physical collection and their digitized data, but this entire ecosystem of potential types of data, both primary, things you can get directly from the image, something like morphological data or a tissue sample, so like this tertiary data that include things like the overall species distribution, a phylogeny, even microbial composition we're able to get from herbarium specimens. And I think this is a really powerful way to think about herbarium, herbaria, and their role in current modern biological research, which is incredibly integrative and interdisciplinary. And it's been a really fun way for me to approach my research program here at LSU. And so I'm going to be doing quite a bit of bragging about the wonderful lab I've established here at LSU over the last few years. We don't actually study Louisiana plants too much quite yet. A couple little side projects getting off the ground, but we really focus on plant evolution in Latin America. Use this extended specimen network sort of ethos to approach our questions, which are right at the intersection of macroevolution, so studying these big patterns of how species came to colonize different areas of the world, how they've diversified, how different traits have evolved, with microevolution, sorry about that, how patterns of gene flow happen between populations of species and what's going on at sort of the more finer scale, and species ecology. So we're really interested in how pollination syndromes and ecological niches intersect with macroevolution and microevolution. So we sit right at this happy place. But everything that my lab does is based in this overall concept of, or this overall framework of systematics. So everybody in my lab is working hard to understand their group from a taxonomic perspective, including a phylogenetic perspective. So we support a lot of alpha taxonomy in the lab, in addition to big phylogenetic projects. So I'll talk to you a little bit today about what Diego and Laura and Lamone and Janet have been up to, and a little bit what Daisy, Eleanor, and Matthew have been up to as well. So for those of you who are familiar with my research, you know a lot of it has used this amazing system to understand these questions and macroevolution, the Neotropical bellflowers, just a beautiful group of plants with these gaudy, fantastic flowers that have generated a lot of exciting questions. A lot of these relate to evolution of pollination syndromes, with these brightly colored flowers being pollinated by hummingbirds and these little bit more pale colored flowers being pollinated by bats. And how does this happen? But we're also interested in the impact, for example, of Andean uplift on driving diversification and how these plants have colonized Latin America sort of more broadly. And like I've been working on this group since I was a PhD student, but I want to give a shout out to Lauren Frankel and Janet Mansouri, two members of my lab, one now PhD student at University of Wisconsin, who have carried on this research and are doing really exciting stuff with it. So we've used the Neotropical bellflower system to sort of establish this extended network, extended specimen network type research program. We start at the very base and understand taxonomy and systematics, how these groups are related. Here's a beautiful illustration by Bobby Angel of a species I described in 2015 or 16 called Cyphocampylus siberiensis. And without study of herbaria physical and also digitized images of herbarium specimens, I wouldn't have the taxonomic knowledge necessary to be able to identify this as a new species and put it into the its overall place within the classification of this group. We also take morphological data from specimens, not only to understand alpha taxonomy, but also to do these comparative studies to understand, in this case, how pollination syndromes, hummingbird in pink and bat in green, sort of map out in morphospace. We use that latitude and longitude data that is often included on the label data of a specimen to subsequently infer species distribution models, like this one for a central pogon species that's endemic to sort of Mesoamerica, trying to understand both where it might occur today and project both into the past and into the future to understand how its range might shift as climate changes and things of that nature. We also pair field work with understanding of morphological variation within this species to try to understand its context. So in this case, the different lengths of these stamen in the field, I was able to show in Bolivia, where these two species occur, I was able to show that this is relevant to where pollen is placed on the back of their hummingbird and bat pollinators. And by looking at the differences in the lengths of the stamens across the specimens, we saw that when these two species, Centropogon mandonis and Centropogon bretonianus, co-occur, the differences between their stamens are really more exaggerated than when they do not co-occur. So this is consistent with this evolutionary pattern called character displacement, which probably allows these two closely related species to co-occur. And finally, I'll talk quite a bit today about how we're able to get phylogenomic data. So we extract DNA from specimens that can be really old, collected back in the 1940s or maybe even before, to generate this useful data that we can use to improve our knowledge of relationships in this group. And so I've most extensively worked with this group, the Neotropical bellflowers, but my lab has been quickly expanding to look at other groups as well. Together, Daniel and Laura are tackling two Neotropical tree groups, Otoba, nutmeg relative in Myristicaceae, and Fresiera, a tea relative in the Pentaphylocaceae. Lamone here is working on the tribe Hilii in Rubiaceae, trying to understand patterns of pollination syndrome in the context of phylogenomics. And my PhD student, Diego Paredes Berneo, is working on this high Andean group of melastomes called Brachiotum to try to understand how Andean uplift has driven rapid speciation and potentially facilitated hybridization in this group. And all of these projects rely heavily on study of herbarium specimens and phylogenomic and morphological data derived from those. But as I mentioned, all the research we do in the lab is really rooted in alpha taxonomy and a good understanding of systematics of the group. So I just want to give a shout out to Daniel Santamaria Aguilar, who's a fantastic generalist botanist who's based in the herbarium. And here he is in La Selva, Costa Rica, holding a fruit of a Varroa species above a figure from a taxonomic synopsis he wrote of he wrote of this group, which involved a ton of just studying the morphology and variation in specimens. Laura and Daniel have been working together to understand the evolution of another nutmeg group, this time in the genus Otoba. It's a really abundant group in Western Amazon. They're not particularly species rich. And Laura was able to generate the first molecular phylogeny of this group using DNA extracted exclusively from herbarium specimens. But she didn't stop there with her use of specimens. She then took latitude and longitude data, and undergraduate Daisy Singletary, who's watching, hey Daisy, she was able, Laura and Daisy were able to create these species distribution models that do a fantastic job of showing just how labile evolution of the niches in this group. So you can see this clade that includes Otoba glycocarpum parvifolias all over the Amazon basin potentially, while other species like Otoba gordonifolia are located just to a really small strip of South America. And this small strip, for those of you who know the Andes, who know South America, know that those are the Andes Mountains lighting up. So we also just looked at phylogenetic comparative methods of that same climate data. We're able to show, yep, the niche evolution has been really dynamic, even though biogeographic movement hasn't been particularly extreme in this group. Laura and Daniel are also working together on the genus Fresiera, and Daniel's described many species in this group and worked on many different treatments. And Laura was able to take a combination of both herbarium and field collected samples for the first densely sampled phylogeny of this group. And even though it's a taxonomic nightmare and it's been sort of ignored for a long time, in part because it is so complex, Laura was able to resolve it into, I think, 6 well-resolved clades. And she paired that herbarium-based phylogenomic research with morphometrics of herbarium specimen derived leaf shape. And here are just outlines that come directly from herbarium specimens of Fresiera, showing, if you just stand back and look at this, it's a pretty impressive variation for a single genus that occurs all in one biome, with 400-fold variation in leaf size, varying from very small and circular and things like Fresiera janajagensis to species that are much longer and have grip tips across the group. And she's done a lot of great work further using this morphological data to understand how this correlates to environment. So more on that soon. We're also interested in what the impact of phylogenomic data is on, herbarium DNA is on phylogenomic inference. And here in Otoba, Laura has shown that there is definitely an impact of sort of where the specimen was collected on the quality of the data we're able to get from it. So Otoba is a group that's native to the wet tropics, and in the wet tropics, it's really common for species to either be collected in ethanol so that they don't start decomposing before you're able to dry them, or maybe you think you're going to be able to dry them really fast, but life is just so fast in the wet tropics that mold can begin to grow really fast. And so that compromises data quality. And Laura has shown that that is actually true, that there is an impact of the environment where an herbarium specimen was collected originally on the quality of phylogenomic data we're able to get from it. And then a former undergrad, Lauren, was able to show that just overall, sort of averaged across all herbarium specimens, we included in an analysis of the bellflower clade, that herbarium data is generally lower quality than data collected from silica specimens. Here you can see that the mean length of the loci that we are able to get from herbarium specimens is shorter than those from silica or field collected tissue, but still fine. We're still able to get a lot of data from them. So moving on to the next project, Janet and Laura have been working together to understand the coevolution of the sickle-bill hummingbird, this obligate pollinator with this super curved bill, and there are 2 groups of food plants, subclade of Centropogon, the Neotropical bellflower group, and Heliconia. This project has evolved a lot of niche inference, phylogenomic analysis from herbarium specimen DNA, and we're also expanding out to include analysis of pollen from the head specimens. So here we have a really fantastic collection of vertebrates that complements the herbarium. That includes one of the largest collections of neotropical birds, which has worked out really well for us. So we are able to go into the weird head divots of the sickle-billed hummingbird and get pollen from them. And so that's a really fun integrative project looking at a group that we already have a pretty solid sense of how biogeography and macroevolution have proceeded. Now we want to understand how pollination syndrome and networks influence the microevolution in this group. And Laura has done some great work looking on sort of establishing a solid hypothesis. We're hoping to field and test, but we have
Laura, are you still there?
Oh no.
Okay.
I am. Can you hear
Let's
me?
see. It seemed like you were starting to get kind of pixelated, so I was going to see if maybe you could
Okay.
just stop your camera, but you could keep talking. That
I
might
can do
help a
that.
little bit.
That sounds good. All right, let me share this again. Oh, that's the wrong view. Sorry about that. I'm on a
No worries.
Wi-Fi connection, not a wired one, which I think sometimes goes wonky. Okay, so yeah, Laura's done some fantastic morphometric data to show that these two species that co-occur and we think have different pollinators, Centropogon granulosus with this very curved flower and Centropogon solanifolius, the straighter flower. She has used herbarium specimen, flowers from herbarium specimens to show that where these two species do not overlap, they have overlapping morphology, they're similarly curved. But when these species do co-occur and those yellow dots at those mid elevations, we don't have any overlap in floral morphology. And so again, this is another potential signal of character displacement that we'll hopefully be able to go out to the field and test later this year or in 2022. And then finally, the last project in my lab that I wanted to highlight is a project that integrates machine learning and pollination biology in the group Hilii and the gene, including the genus Hilia. Here, my graduate student, Lamone Ball, is really interested in how Hilia has become so florally diverse. Despite having only 24 species, it has flowers that are hawk moth pollinated, bat pollinated, and hummingbird pollinated. So we've partnered with the LSU Center for Computation and Technology. Here I want to shout out to Dr. Lyndon Coghill and Ilham Ravankabash, who have played a major role in establishing a neural network that takes herbarium specimens and is able to accurately predict pollination syndrome from that using a test data for that species for which we know the pollinators. So this is a really fun tool that integrates herbarium specimens into this very sort of machine learning framework that is pretty exciting in biology right now, doing a lot of rethinking of how we approach biology. And so that's what we've been up to as a lab. I'm really just proud of my lab for using herbarium specimens in such creative ways and generally for doing such excellent research. But we facilitate much more than just my lab's research at LSU. And so I wanted to highlight some of the other research that we've helped to make happen. Here, Jenny has already mentioned Dr. Christopher Reed. So Chris Reed is an excellent botanist of Louisiana, probably knows the flora here better than anyone. And for his PhD research, working with my predecessor, Lowell Urbach, Chris Reed inferred phylogeny of a group of sedges shown here, and has also paired this with really fantastic restoration efforts here in Louisiana, working not just on sedges, but generally on some of our more threatened ecosystems. And he also has added to the collection via both international and domestic fieldwork. Dr. Kyle Harm's lab at LSU also uses their collection. They both use it to identify the specimens that the species that they study, mainly in the longleaf pine savanna. But they also add to it via vouchering the specimens that they collect in their research. A lot of their lab looks at fire ecology and super cool ecosystem down here in the southeast, the longleaf pine savannas, where you can find tons of these carnivorous pitcher plants, the Sarracenias. And so 2 PhD students here are highlighted. Carrie Barker's research is looking at the impact of different sort of shrubby species on the forb composition down below. And Luis Santiago Rosario is looking at, actually not necessarily just in the longleaf pine savanna, but he's collected sunflowers across a environmental gradient to try to understand the impact of salt on species physiology. And both Carrie, Luis, and Kyle are very active members of the herbarium community and deposit specimens there regularly. We always, Jenny mentioned, we have a fantastic collection of fungi here at LSU. And so a PhD student actually working on microfungi in mammal lungs for most of her thesis, but Spencer Biernacky is working doing a little side project trying to delimit species or determine whether this is more than one species, this Microcybes titan, and other fungal research. We were able to help other ongoing fungal research projects in the lab of Dr. Vincent Doyle at LSU as well. And finally, Dr. Sarah Matthews joined LSU, I think, a year and a half ago, and is busy establishing her own research program here at LSU, but she brought with her a really fun project based on the phylogenomics of Australian parasitic plants, including these fantastic flowers here. And it's really exciting to see her get that off the ground and make sure that we can play a role in making sure she can get her specimens from Australia here to LSU and do all the research that she might want to do. And so if there's any way that, for those of you watching who do botanical research, just let us know if there's any way we can facilitate your research, especially if you're working on questions in the Southeast where our collection is so strong. And just to wrap up, I'm a big proponent of this extended specimen network type model. And at LSU, we've done a pretty great job of, this is not a LSU specimen, but of having a really fantastic collection of physical specimens in our 400,000 or so specimen collection, and then digitizing our collection. So we've done a really great job of making our data available to the broad public. And we're probably one of the few herbaria out there, especially of the size we're at, that can say the end of digitization, at least in terms of having an image for every specimen in soliton data. We're one of the few where there's actually sort of an end in sight for us. So it's really exciting. And so in the next years and probably decades to come, I hope that we can continue to go from this very well-established link between digitized specimen and publicly available data to these extended network type data sources, things like posting morphology to databases, your DNA sequences, and other types of data. And here I just want to give a shout out to BRIT, who are hosting us today. So thank you specifically, Brooke and Tiana, but in general to BRIT, because by the partnership that we've had with you, we've been able to really grow our collection and make it a more important regional collection. And that the data we now share is more valuable to anybody who's interested in botanical questions. And so with that, I'm just going to go ahead and wrap up and say thanks. Thanks to all the herbarium students and staff who make sure this collection runs smoothly. Thanks for Elham and Lyndon to being open to listening to Lamone and I blab about our botanical whims. And then thanks to the National Science Foundation for supporting our current integration project, as well as past sort of digitization projects, as well as a recent one focused on bryophytes and lichens that perhaps some of the herbaria that you're at are also working towards. And no talk from the LSU Herbarium can be complete without acknowledging just the amazing powerhouse that is Shirley Tucker. Her donation has made it so that the herbarium is in a really exciting place, and we're able to do a lot of really exciting research. And I thank her, and I'm sure Jenny echoes that. She's just been fantastic. It's been fun to try to live up to her dreams. And so with that, I'll go ahead and hop off.
Thanks, Laura.
That was an excellent talk, Jenny and Laura, and I appreciate you all spending your time with us today. I think you did such a great job about showing how passionate you are about the collections and how they're really dynamic and they have such a place in current, future, past research. And I especially loved how you called out how you really emphasized how herbaria, like moths to a flame, attract all sorts of researchers and botanists to make this really cool synergistic hub, an innovation hub. So well done. Thank you very much. So we do have a few questions in here, and it looks like the first ones are coming from Harold Keller, who is a resident research associate here at BRIT. And I think these are probably directed to Jenny, but please step in anytime. Laura, let's see. So the first one is, do you exchange type specimens with herbaria in Russia?
Good question, but no, I have lent specimens on exchange to a researcher in Russia, but no, I don't have any exchange type specimens from Russia.
Good.
And I think it's difficult to move collections between Russia and the U.S. So I think they block, moving collections into the country. So you have to. So that's why we had to go through Belarus in order to get our specimens there for someone to look at.
Thank you. And it looks like Harold has another one. Is Professor Meredith Blackwell's fungal collection at LSU? Were any of her collections transferred elsewhere?
That's a good question. Unfortunately, they're not at LSU. They're at University of Arizona. So I think that was just the timing. And I think it was because we didn't, at the time her collections were moved, it was before we consolidated the collections. So she's an amazing mycologist that was still, part of LSU's community. And so her collections are not here, but she did a lot of work on fungi, especially that are in insect, I don't think she worked on the zombie fungus, but other ones that are associated with insect species and yeast, and a lot of it's microfungi too.
Thank you. Very good. So we're going to switch directions now. This is a question from Peter Fritsch, and this is probably addressed to perhaps both of you. And the question is, how full is the LSU herbarium space-wise? Percentage, more or less? Are there needs or plans for expansion?
So
I can, go ahead, Laura.
Either way. The herbarium, so we were just thinking about this the other day. We have room to grow by maybe a third or so. So we're maybe 2/3 full. We can go up to about 600,000 specimens. There are no current needs for expansion, and it would probably be difficult to expand our collection because it was recently constructed. Our building, I think as Jenny mentioned, was built in 2002 and takes a big chunk of a floor of a biological sciences department. And it's really centrally located and a fantastic infrastructure that's already compacted. It was built compacted, which I think is really neat for it. And so I think it's going to be, I think that we currently don't need it. And I think that in the future, if we do need to expand our collection, it would be difficult to. That said, we don't want it to be the case, but if other herbaria in Louisiana do have to be divested from their collection for whatever reason, we would have space for them. That said, I hope it doesn't happen because herbaria are so important at institutions. But yeah, we would be able to, in an emergency, help other herbaria. Oh, Anna, you're muted.
Thank you. So this is from Lauren Gardner. It would be great to hear more about how the herbarium is funded. Do you get a combination of funds from the university, research grants, and major grants like NSF and philanthropic funding?
Yeah, all of that. So we are really lucky to have one major philanthropic benefactor in Shirley Tucker. And we have minor donors as well that support our research, including the
McRoberts.
Thank you. The McRoberts family, who, I'm currently blinking at what institution they were at.
They're at LSU Shreveport, so they were the curators there for many years and have retired. And some of their collections have come to us, and some were repatriated as well for the Caddo Parish flora. So they still keep an eye on our collections and support us too.
Yeah, so it's really great. Our department does support us to a certain extent. We get a small operating budget, but it's pretty small. And then we've been really lucky to have NSF support. So we currently have a collections and support of biological research grant from NSF to support the integration of all those different collections. I don't know if you caught it, but we've doubled in size in the last 5 years. So NSF is supporting that work right now, and will probably for the next year or two on that grant. And we have other grants that support digitization efforts. We're also really lucky that LSU, or not LSU specifically, but Louisiana has invested in sort of undergraduate work study. And so we have a lot of, and of course there's a federal work study program, but many of our students are eligible for funding that way. And we end up doing a lot of work with undergrads who are paid for their efforts via work study program.
Wonderful. Thank you both. All right. I'm going to ask one of my own, because when you said collecting expeditions, I got very interested in knowing that there's finite room in which to grow. Can you tell us more about these collecting expeditions and how you choose and prioritize where to collect and who goes along?
I guess in the past, it's mostly been through classes. So classes going out into collecting all throughout, different parts of Louisiana. So that's been how we've grown in the past. But with Laura's research, we're hoping that there's going to be some tropical, plants coming in.
Yeah. Yep. So I will admit, since starting here in 2017, I haven't spearheaded any major collections, but there's certainly plans of it. But my lab will be starting to do a lot more lab work in the tropics to expand our collection in that way very soon. In that case, it's going to be the individual researchers who are working on different projects. But I hope, Jenny and I always talk about dreams, just getting to the field. And so hopefully now COVID restrictions will start to open back up, and maybe we can do some more local collecting.
I love that idea. Maybe we could go along the border and have some joint collecting expeditions.
That would be amazing. I would love that.
So this to continue on maybe what you were talking about, Laura. So this is a question from Peter, and it says, Laura, where do you do most of your neotropical research? Do you have challenges with the fieldwork, permits, weather, etc.?
Yeah. So I'm just lucky to do field work in many countries. So I've worked in Costa Rica, Panama, Peru, and Bolivia. A lot of the pollination research I've done so far has exclusively been in Bolivia. And yes, permits are a challenge. For me, it's been really helpful to partner with local scientists who I know personally. And so having a professional network that's strong has been really important in being able to obtain those permits. But it's such a challenge, and every country is so different. And with the Nagoya Protocol now protecting data from genetic data of biodiversity in different countries, I feel like the difficulty is even stepped up a notch. And so, yeah, I think when you start to do field work, I think now graduate students and people who are just getting started need to really think about cultivating the network and knowing a year or so out, what you're going to need to do to get those permits. But I've been lucky enough to be able to figure that out. Yeah, weather, there's always rain. Fortunately, I don't think I've been super derailed by weather, fortunately, but it does get cold sometimes and wet.
Thank you. So we have another question here from Eric Tepe, and I apologize if I mispronounced that last name. Laura, is the methodological consequences of using herbarium specimens, end quote, work published? ? We're about to dive into a project that will involve a lot of herbarium specimens.
It is not yet, and that's yeah. But I can if you send me an email, I think maybe some of the relationship with the environment is up on bioRxiv. And the part sort of just looking at the overall quality from herbarium versus not is not currently published. It's a paper that, you know, it's been on my back burner for way too long, but I can share that data with you. And I can also say we are echoing, that result echoes a lot of published research, just showing that in general, you can get good data from herbarium specimens, but reads tend to be, or context, assembled context tend to be a little bit shorter. You get fewer long reads out of sequence capture data. I know a little bit about Eric's research, you know, so it's very likely sequence capture. I would say that the targeted sequence capture method is robust to low quality DNA. And so you'll probably have a good success unless it's really bad DNA extraction, which tends to happen in things, you know, that were collected a long time ago or were collected in ethanol. Being collected in ethanol is just like, it's real tough to get good DNA after that.
Thank you. So here we've got a question from Weston Testo. Hi, Laura and Jennifer. Nice talk. What are your priorities for collection growth looking forward in terms of geography and taxa? What does the future of exchange and collecting look like for LSU?
Yeah, I think because of this repatriation project, it made us realize that, you know, it'd be nice to represent Louisiana. So thank you, Tiana, for really helping us, you know, go through. We sent you a checklist that was the most recent checklist of Louisiana flora by Charles Allen. And we also sent a checklist of rare species for the state. And so with this digitization project, I think we'll get a better handle on you know, how we represent species for the state. And, you know, it also will give us a nice idea of, like, I would like to know, like, where our collection holes are in the state. You know, with all this digitization, you can really look at where, you know, what's missing and have a more targeted collection expeditions rather than just, you know, collecting the same stuff over and over. So as far as the state goes, we're really interested in, you know, keeping going there. But
Absolutely.
But then with you know, Sarah, and, you know, a lot of our collection is like who the curators are of the collection and what their work is. So that's the fun part about an herbarium, is you get to kind of see that history of the collections of, you know, what research has been done. And
absolutely. So. Yeah. And so I just want to echo, like, Jenny, and herbarium strength, what our role in the botanical world is to house collections, plant life of Louisiana and the Gulf South more broadly. We're really strong in that. But that's not going to stop me from doing my own research program, which we're hoping the next year or two will take us to different lab members to Costa Rica, Peru, and Colombia. So there will continue to be sort of a montane neotropical focus in my lab. But in terms of collections growth, I think it's good to have sort of global representation as much as possible, but I think it's most important for the bottom line of our collection for the growth to be regional as well.
Thank you both. Those are fascinating answers and the sorts of things I think we're all wrestling with. So it looks like we've got one final question in here, and it goes back to Laura. Do hummingbirds and bats ever pollinate the same flower species?
Yeah, Harold, they absolutely do. And so I don't know if you remember, but there was a slide where I had a red flower and like a yellowish flower. So that's just one instance where those two species share both bats and hummingbird pollinators. The red flower in that slide was mainly hummingbird pollinated, but there was an important role of bats as well. I think like 20% of the pollen transfer was from bats. But the other flower was very much generalist. It was almost approximately 50-50 of the pollination heavy lifting was done by hummingbirds and bats. And so, yeah, a lot of research in my lab tries to understand why would you go through this generalist phase? What are the evolutionary and ecological pros and cons of that? So absolutely.
Thank you. And I think that's our last question in there, but I just want to echo the many thanks that appear in the YouTube channel as well. We appreciate y'all spending the time with us. Many of us in herbaria don't always work with, large groups. And so it's really nice to hear the stories, hear your passion about it, and hear how you do things. And congratulations on all the hard work that I know you put in every day and all the cool stories you're able to pull out of the collection. So thank you both very much. And thank you to our audience for listening.
Yeah, thanks everyone.
Thank you. Thanks, Tiana and Brooke and everyone.